message box provided in the application. And also try to mute your microphone during the presentation to prevent any background noises. Are there any questions? Okay, so our first presentation will be presented by Brian Bott. Brian received his bachelor's in statistics from the University of Minnesota, Twin Cities. Brian worked in the Department of Biomedical Statistics and Informatics at the Mayo Clinic for 7 years. He is currently a senior scientist and a community manager at Sage Bionetworks, which is a nonprofit organization dedicated to exploring open source models in the advancement of biomedical research in Seattle. And today, Brian will be presenting about Synapse, which provides a private or public workspace that allows you to aggregate, describe, and share your research. Without further ado, allow me to introduce Brian.
Great, thank you. It's nice to be on the call, and it was nice to put faces to a lot of these names as well down at UCLA last month. So I gave a short overview at that meeting for those who weren't at the face-to-face. I'll give a brief overview of the platform, about Sage Bionetworks in general, and then I'll give a little bit of a live demo as well to really drive home how you would actually use the tool to use Synapse. So as was mentioned, Sage is a small nonprofit. We're located in Seattle, Washington. Our offices are in the Fred Hutchinson Cancer Research Center. We're about 40 people, about half of whom are staff scientists, about a third of which are software engineers that build a few different software platforms, one of which is Synapse, and then the remaining portion of the organization is leadership and support staff. So Sage is focused on a world where biomedical research is going to fundamentally change to be more open and collaborative. That's sort of the baseline where we start from. We believe so strongly in that all the work that we do is sort of built on that assumption. We pilot a variety of components that we feel are necessary to build an open scientific commons, and in doing so, we support a number of communities of researchers who work collaboratively across research disciplines. And Synapse in particular is a platform to support open scientific collaboration and enables sharing of digital resources, data, code, results, and importantly, their relationships to one another. And it really provides a mechanism for people who may not be co-located to be able to communicate in real time as opposed to at time of publication. So importantly, there are lots of very, very good tools and technologies out there. We're not trying to reinvent a lot of them. We're trying to build layers on top of them that make them more useful and integrate them. So, for instance, GitHub is a great model, a great code versioning and social platform. We use it internally at Sage for most of our code development. Amazon Web Services provides great cloud computing, as does Google Compute. We leverage a lot of those compute resources. So all of the tools that we provide through Synapse really can still leverage all of these great tools. And what we do is help co-locate them and describe their relationships to one another. So Synapse is built entirely on open RESTful APIs, such that all of the content within the system can be accessed not just through the website synapse.org. That's one way to interact with Synapse, but you can also interact with it programmatically through a number of our analytical clients. We have fully supported R client, as well as Python and command line clients. And when you really boil it down, Synapse is a distributed inversion file system where you can richly annotate, provide metadata on top of files that you're sharing with operators. We have the concept of web-accessible tables, which I'll show here shortly, where you can provide customizable schemas for very structured data to allow queries. And we're also agnostic to a storage solution. So Synapse provides, again, this aggregation of resources. By default, we store things in Amazon S3 for things that are quote-unquote natively stored in Synapse. But I'll show an example too of essentially just being able to reference something that lives elsewhere on the web. We provide wiki authoring such that you have the ability to craft a narrative right next to the files, the data, the code that you're sharing. And importantly, we have access controls such that you can share whatever you want with whomever you want. And those access controls are dynamic. So over time, you can add more people as the collaboration may grow. And then, for instance, make a project public once you publish. So because we work primarily in biomedical research, we also have, on top of access controls, we also have a governance process. So all users agree to certain terms and conditions, essentially saying that they understand basic rules about, for instance, HIPAA and identifiability of data, but we also provide another level of governance on top of that, where if there is more sensitive data that may require other types of permissions, for instance, an IRB approval, we actually have the ability to layer on top of these web accessible files another layer that a user would have to go through in order to access them. So that's a real brief overview, and I think now I'll jump over to the website. I'll break real quickly here if people have high-level questions. Okay, great. So this is, let me actually log out. So coming to Synapse as a new user, you can log in, register. Accounts are free. We are a nonprofit. We're funded through a variety of mechanisms, including philanthropic donations, funding through NIH grants like the BD2K, as well as other research grants. And our goal is to continue to make Synapse freely available to the research community. So once you log in, you get routed to your personal dashboard, which includes information about yourself, as well as all of the projects that you have access to, and all of the teams that you're a member of. So here you can see the list of projects that I've been working on, ordered by most recently created. I have a list of favorites that I have keep tabs on. These are projects where I may not be, for instance, actively working in them, but I may want to check in from time to time. And I can filter by the team that I'm a member of. And you can see on the Teams tab, these are all of the permissions teams that I'm a member of. So why don't we start by actually creating a new project. So a project is sort of the organizing structure that that all resources are made available. You can create folders. So I can have a data folder, a code folder, and maybe a results folder, just like you would on your local hard drive. Importantly, any resource within the system has the unique identifier, and that identifier allows you to access that resource across clients. So I mentioned before the concept of an analytical client. Excuse me. So this new project that I just created, I can actually copy that ID, and if I go over to an R prompt, for instance, I can load the Synapse client there, and I can call synget and retrieve that object. So that object is a project. This is the ID. This is where it lives. If I had included any annotations on it, that would be accessible as well. So it really provides a way to dynamically create and consume content, both interactively through the web as well as through programmatic clients. So I may want to add an annotation to the project and say site is UCLA or something like that. So now if I go back over to the analytical client and do that same Synapse get, and I take a look at annotations of project, you can see that, that annotation there. That also allows me to query across the system. So I can do something like Synapse query, select ID and name from project where site is equal to UCLA. And this will return the project that we just created. So it really provides a powerful tool, again, to be able to both create and consume content in a number of different ways. You can jump over to the wiki. It's a pretty simple markdown-based editing system. BD2K demo. This is pretty neat. All right, guys. So you can quickly craft narratives as well. I may create another page, additional instructions. You can start to build up a hierarchy of wiki content. Then we also have, I mentioned, the more structured tables. There's documentation on which you can read about in our Getting Started guide. But what I'll actually do is I go to an already created table so you can see the types of things you can do within a structured tabular format. So I showed you how you're able to query within the system across objects, so you can search for all projects with a certain annotation. What these tables allow is for you to query within the content of a more structured table. So this is a table that I've created previously. We can take a look at the schema. So there are 4 columns. These are the column types, and for the strings, we have different sizes. And what this allows me to do is to write SQL-like queries, such as I'm only interested in rows where treatment arm is A, for instance. And that's returning only the rows where that condition is true. I can take that entire query and go again to the R client and say table query. Copy and paste that right in there, and that's going to run the same query and return that data in, again, a structured way. So this is the exact same start of that query that we see in the web portal, but now we have it in a DataFrame, which is loaded directly into your analytical environment. So you can start to do an analysis based on only a subset of things. This is nice in general. Where it becomes very powerful is when you can also start to include other types of columns. For instance, you could have a column that is a file. So you could imagine having a demographics table like this with other columns that are, for instance, gene expression array or BAM files or other information that would be very important in linking to a sample, for instance, and that would allow you, again, programmatically to go in, extract only the individuals that you're interested in, such that you're not retrieving all of the larger blobs, the larger files that may be associated with that. So I think I'll probably just do one other small bit to show that information doesn't necessarily need to live completely within Synapse as well. So I created these folders in the demo project, and I'm going to grab the ID of this code folder and go back over to the R client. And I have a real short script here, which is loading a few other packages and sourcing in a function that is out in GitHub called knit to Synapse, which all you need to pass it really is a file and a place for you to put the output of a knitter script into a Synapse Wiki. So I have this folder that I just created. I'll show you shortly where this hello world script lives. But what I'm doing here is I'm retrieving the URL to a specific commit of a file stored in GitHub. So I'll show that here. So it's at raw.github.com, sage, say Synapse demo, a specific commit, and the path to the file. And then I am registering that with Synapse. And then I am going to write wiki page back to Synapse with the output of my knitter script. So I'll come back to the web here. I'll refresh this page. And now there should be a file here called hello world. You can see that this doesn't live natively in AWS. It's an external storage. If I click on this, this is the web URL. This is what the knitter script looks like. So something real simple. It's a narrative with inline code. Tell me that pi is equal to, and you execute that, and the plot to show a simple function. So if we go back to the Synapse page here, you'll see that there is also a wiki attached to this file with the output of that script. So very quickly you can consolidate analyses, consolidate work that you're doing, and in this example, really be able to share that across collaborators. So I think I'm at about 25 minutes here. It might be good to open it up for questions or comments.
Thanks, Brian.
So this is Andrew Sue here. Brian, cool demo, thanks. I'm curious, right? I mean, are there example projects that are public that we can go, look at that essentially show what this type of environment can grow into?
Absolutely. Let me actually go to some of my favorites. So this is the private project. Let me search for AD Knowledge Portal. So this is a good example. So this is the Accelerating Medicines Partnership funded through the NIH, the FNIH, and the National Institute of Aging, where there are 4 academic consortia who have come together to generate a large bolus of gene expression and genetic information, specifically in Alzheimer's disease. So the goal of the consortium is specifically target discovery and preclinical validation. Every 3 months there will be data releases through this project. At this point, it's very early. We've just had the first data release last month. But there is lots of information about the data that's available. What will be coming in subsequent months is a little bit more of the analyses that are built on top of this. But here you can see the sharing dialogue here. It's open to the public. Anybody can go to this. And this is one of those structured tables that provides information about all of the different types of data that is included in the project. So I think this would be a good resource. I believe there's a link to that.
And just to confirm, this table, I'm not seeing it on.
Say that again?
Well, so I found the AMP80 Knowledge Portal. I just want to confirm that the tables browser that, oh, there it is. I missed the link, I think. Okay, got it. I see the master table now. Okay, cool.
Okay.
So, I mean, I guess, I was also, since you mentioned, I was cruising through the files, and it seems like this right now primarily serves as the data centralization point. Is that accurate? I see, there's the Broad, Emory, Mount Sinai, all of whom who have data subfolders.
Yep.
So I guess I'm wondering about, collaborative analyses, right?
Yeah.
How that works, because that, I would imagine, is one of the big selling points here.
Absolutely. And what I can show you is briefly the private project where these groups are currently carrying out their collaborative analyses. So if you can see this, unfortunately, this isn't public. This isn't something that you have access to at this point. But as these analyses get vetted and published, it will essentially get published through Synapse as well. So you can see the different types of collaborative analyses that they're undertaking, all the different proposals here. And again, there's folders with collaborative analyses, as well as they store meeting notes from all their meetings. So it's a growing resource and something that they're being tasked with making their data publicly available every 3 months. I think they're a little bit more nervous about getting scooped and making their analyses available in real time. But I think making the data public is making them work very quickly to get some of these analyses published, and we'll have better examples of you know, a collaborative environment out in the open.
Cool. Yeah, I mean that's what I would be really interested in seeing, right? In terms of you know, how people interact, you know, between distributed teams, Synapse and GitHub, for example, because I can imagine, you know, that's a big challenge with any distributed team.
Absolutely.
Great. Thanks a lot. Cool.
All right. Well, thanks everybody.
Thank you.
Thank you, Brian. Okay, so next up, our second presentation will be presented by Ali Torkamani. He received his bachelor's degree in chemistry from Stanford and his PhD in biomedical sciences from UC San Diego. Ali has worked at Scripps for about 7 years as a research scientist, faculty, and now as the director of Genome Informatics at the Scripps Translational Science Institute. Today, Ali will be presenting about the Wellderly Cohort, which seeks 2,000 elderly volunteers to donate their DNA to the program's gene bank. And these samples are studied by Scripps researchers to uncover the protective elements of the human genetic code. Without further ado, allow me to introduce Ali.
Thank you. Can everyone see my screen okay? Can you see my slideshow?
Yes, looks good.
Okay. All right, so I'll be talking about the Wellderly Cohort, as was described. This is a resource that we've built at the Scripps Translational Science Institute for as a kind of healthy aging cohort. I'll get into what the inclusion requirements and all that are for this cohort in a later slide. But first, I wanted to just briefly mention a little bit about what we do at Scripps. So I run most of the genomics programs that we have going on at the Scripps Translational Science Institute, and we have programs kind of across the spectra from pre-birth to death, really, using genome sequencing in various different ways to look for, you know, genetic markers of these various kinds of conditions. So everything from analysis of non-invasive prenatal testing type of test all the way to molecular autopsy, which is sequencing of individuals who died unexpectedly and are trying to find a cause for their sudden death. So I primarily focus on building interpretation tools for this sort of analysis. I mean, really plan the entire study and all that, but then focus on actually, once you have the data, what do you do with it? So we've built this tool, Scripps Genome Advisor, available as a web service for variant annotation. You can think of it as something similar to the myvariant.info that Andrew, I believe, presented, or Chun-Li presented previously. And then we have a commercial version of the software as well. So with that, I'll be focusing now on our particular project that we're interested here for this consortium, the Wellderly Cohort. And internally, we're interested in the Wellderly Cohort for the identification of variants, protective variants in particular, that may explain the Wellderly phenotype. So these are individuals over the age of 80 with no common chronic diseases. We have genome sequence information on a subset of them. I'll get into the details of how many individuals have sequence and of what type, etc. But the overall idea is that there's something special about these individuals in terms of some genetic explanation as to why they live to or beyond the age of 80 and don't have any of the common diseases that you see across the general population. And so we're in the process of aggregating, a slow, painful process of aggregating the information that we have across all these individuals to do our statistical analysis. And what we'll be pursuing internally is a combination of annotation plus rare variant analysis to identify sets of rare variants that explain this phenotype. So you're all probably familiar with genome-wide association studies and single marker tests. This has been done in some healthy aged populations, and there's a couple of weak markers that have been identified, but nothing stunning. So our hypothesis is that perhaps there's rare variants that are protective, and I'll show evidence later that there doesn't seem to be any evidence for a lack of disease-causative variants. So this statistical approach that we'll be taking, we've sort of developed a proof of principle here. It hasn't been published yet, but we're in the process of writing it up, where we're taking an annotation plus rare variant analysis approach to response to clopidogrel, an anti-platelet agent, where we sequence, whole exome sequence, 350 individuals from a larger cohort, shown in the top left kind of histogram chart, where we have these individuals in blue, and it overlaps with the green, that have been assessed for platelet function after exposure to clopidogrel. It's actually an in vitro assay. Then selected extremes from this distribution, shown in green, and then went through a series of statistical analyses. So when you do the standard simple locus analysis, shown on the bottom left, you get this peak, like 52C19, which has been described multiple times previously. And so then from there, the set-based analyses, what you do is use annotation to prioritize rare variants that are more or less likely to be functional, combine them into gene-based sets, and then there's a series of gene kind of burden-based statistical tests that you can apply. And we identified another P2 receptor. So clopidogrel acts through a P2Y receptor, and we identified another P2X receptor that is statistically enriched with rare variants that differentiate responders from non-responders. So this is kind of a proof of concept that this approach seems to work, and we're hoping to apply it to the Wellderly. So with that, I'll actually get into what the Wellderly cohort is and how it can be useful to the BD2K consortium. What are the resources exactly that are available? So this is a more detailed list of inclusion criteria. The basic inclusion criteria is over the age of 80 with no common chronic conditions, but there are some exceptions. Really, the exclusion criteria is listed here. They're allowed to have skin cancer, for example. So there's actually some conditions that are actually allowed in the cohort. These are cancer, diabetes, heart attack, stroke, autoimmune disease are really the exclusion criteria. There's a much longer list of ICD-9 codes, but this is sort of what it comes down to. And the total cohort is about 1,600 individuals. It's probably slightly above 1,600 individuals right now, and we have some clinical phenotypes on these individuals. They haven't been, I wouldn't say they've gone through a kind of rigorous clinical screening at Scripps. That hasn't been done, but we have some information from their medical records anyways. So here's the breakdown of the cohort as compared to the U.S. population. So their median age is 82.9 versus 37.2 years in the U.S. population. The other comparisons that I'll make are actually to U.S. population that's 80 plus or 70 plus years old, depending on what data was available. So the gender distribution is about 60-40, female to male. That's approximately equal to what you see in the U.S. Population over the age of 80 years old. Their median heights are shown here. They're about the same height as everyone else. So there is an association between shorter individuals tend to live longer, but there doesn't seem to be any selection for height here. They do weigh less than the average individual of their age in the U.S. Population. But there appears to be no difference in smoking, maybe even a little bit more smoking in the males. They do exercise more, it appears, but whether or not that's due to the fact that they're able to exercise or not is unclear. And then the education distribution is shown here, so they tend to be more highly educated than the individuals in the U.S. population at their age. So there's some evidence maybe for non-genetic factors that could be driving this phenotype, but I wouldn't say anything terribly overt. But we kind of wanted to determine, well, can we come up with any other evidence to kind of determine whether or not this is a genetic phenotype or not? The one analysis that we have done is looked at a survival curve for the siblings of the Wellderly individuals versus 1920 birth cohort. So we started recruiting these patients a while ago, which is why they're compared to the 1920 birth cohort. And so their survival is shown in blue, the Wellderly survival is shown in blue, and the U.S. population survival curve from 1920s birth cohort is shown in red, and there's a statistical increase in their survival of the Wellderly siblings. A couple things to note is, one, I started the survival analysis at about 5 years of age. We're not sure if we have accurate like infant mortality for this cohort, and there is, if you started from 0 years of age, there's a big drop in the 1920s birth cohort of infant mortality that we didn't see in the Wellderly siblings. So we started from 5 years of age, and you see they don't really necessarily live longer than individuals from the 1920s, but they tend to survive this middle age period better than the 1920s birth cohort. Whether or not that environment versus genetics is still unclear, but at least some evidence that there's something interesting going on. So here's the actual resources that we have available. So 500 of these individuals have whole genome sequencing on the Complete Genomics platform. 200 of them have phased whole genomes on the Illumina molecular platform, 100 of which overlap between these two cohorts. So it's 100 that have both Complete Genomics and Illumina data, and 100 that have just Illumina data on its own, and then 400 that were approximately 400 that have only Complete Genomics whole genome sequencing data. We also have genotyping data on about 1,000 individuals done on an Illumina array, and we're in the process of generating IPSC lines for this cohort. The target is 75 individuals from this cohort will have 3 IPSC lines generated per individual, and these individuals have been, so these lines are being generated for another a project looking at 9p21 and the kind of molecular mechanism that 9p21 drives risk for various cardiovascular diseases. So they haven't been selected necessarily for whether or not they have genome sequencing data or not, but about 50% of them will end up having whole genome sequence data. And currently, we have 11 individuals with IPSCs, with Complete Genomics genome sequencing data, and 4 individuals that have both CGI and molecular whole genome sequencing data. And then the lines are at different kind of stages of validation as well. So 15 of the lines have been kind of fully validated in terms of being pluripotent, and so we check for the pluripotency markers as well as whether any chromosomal abnormalities show up in the lines to make sure there's no just aberration in these lines. So 15 of the 28 that we have in total have been fully validated. So as a cardiovascular disease cohort, there's these opportunities sort of available from the IPSC lines. We could derive smooth muscle cells and endothelial cells and characterize them and do functional assays, etc., and sort of have that available alongside with the whole genome sequence data. Some of these individuals will be undergoing this process of generating these derived lines, but it's not going to be done in any systematic way, so we have to think about whether or not we want to do that. There's obviously the whole genome sequencing data. Another thing that we discussed at our meeting at Scripps is to do RNA-seq on these derived cells and pair that with all the other information we have. And finally, we talked about doing proteomics from the blood. So I looked into that as well, and the requirement that I got was blood and sodium lithium tubes. And it turns out we do have blood, but it's been collected on sodium heparin tubes. So I don't know if that's an issue or not for the proteomics analyses. But if it is, you could always apply it on the derived cells instead. And then I thought, in terms of bioinformatic resources, I might lay out some potential possibilities as well. Some of these things are already happening. So within Scripps, I mentioned we have the Scripps Genome Advisor annotation pipeline that my lab has built, and the Wellderly allele frequencies are distributed through that server. There's a they'll also be included in the mygene.info and myvariant.info pipelines once it's been condensed by the SDSC. So we also have a collaboration with the SDSC to take all this data and kind of normalize it, combine it across the cohort, and generate a web server that we can use for accessibility of this data. So when that's done, we input some of this allele frequency information into Andrew's resources. We could build a beacon for this web server, and the other thing that's coming out of the SDSC collaboration is a web server for query of the Wellderly data at the aggregated variant level, similar to the exome sequencing server, if you're familiar with that. So I sort of talked about this already. So just very briefly, the variant frequencies that we compiled ourselves are already released and available at this URL. There's the Scripture Advisor URL. And the data has been used in at least one publication so far, showing that the Wellderly have a decrease in the frequency of this mutation that's been identified as a risk for Alzheimer's disease. I'm going to skip over the too much of the detail of this web server database that we're building. But the ultimate goal is to combine data across these two platforms, normalize the variance so there's inconsistent representation of variance across two platforms, or even within the within a platform across individuals. There's inconsistent representation of variance, so that needs to be fixed. And then the other challenges are making this large data set queryable externally as well as internally, so that we can do kind of on-the-fly statistical analyses, and that's being done using this Vertica database resource. I'll skip over the details of this. I don't think it's all that interesting, really, on the computational side. So here's another initial analysis that we've done for this cohort, looking at whether or not there's disease-causative variants in this cohort. So one hypothesis might be that they have a decrease in the number of disease-causative variants. Defining disease-causative variants by itself is a little bit of a issue. So what's shown in this pie chart is the entire pie chart is all variants in the human gene mutation database that are annotated as disease-causative. If you're at all familiar with the HGMD database, you'll know it's full of false positives. Estimated that about 30% of the entries in there are false positives. So it's broken down, this whole circle is broken down by variant, the classification of these variants in ClinVar, which is supposedly a cleaner, expert-driven classification of variants by pathogenicity. So most of the variants aren't classified at all in ClinVar. And a small slice, these 32 variants out of the total few hundred variants are classified both as pathogenic in HGMD and ClinVar. So when you use that number as the number of pathogenic variants in the Wellderly cohort, that comes out to about 5% of the individuals having a pathogenic variant in one of these genes that's deemed reportable by the American College of Medical Genetics for incidental findings. So it's a small list of genes, about 57, I think, genes that are deemed highly actionable by the American College of Medical Genetics. They generally drive dominant conditions, and most of them are cancers, either inherited cancers or cardiomyopathies, is the majority of these genes. Anyway, this 5% figure is about equal to what you see in other cohorts that haven't been selected for kind of healthy elderly status. So there doesn't seem to be any evidence that they have a lack of disease-positive variants at this level anyway. We've also taken a look at risk variants, GWAS risk variants, in collaboration with 23andMe. There doesn't seem to be any evidence for a decrease in the frequency of these risk variants in the Wellderly cohort versus the 23andMe population, except for at ApoE4, there's a pretty strong decrease in the ApoE4 allele frequency in the Wellderly versus the 23andMe cohort. So they do have a decrease in this very strong risk factor for Alzheimer's disease. And then finally, to do this, so it doesn't appear that there's a lack of disease-positive variants. That's why we're sort of going with the hypothesis that there's potentially protective variants in the cohort, and we've started a collaboration with the Innova Translational Medicine Institute to see if we can do this analysis. We need a control cohort, and it turns out that they have about 1,000 individuals sequenced on the Complete Genomics platform at approximately the same time. So in collaboration with them, using their cohort as a control cohort, we'll be going through this annotation and rare variant analysis to see if we can find any protective variants. And finally, I'd say that this web server resource that we're building with the SDSC, the ultimate goal is to make it usable not only for the Wellderly analysis, but also for our other genomic analyses as well, where ultimately what we would like to do is have it be kind of a warehouse for all of our genomic data that can be integrated and normalized against one another, and either made available, if it's acceptable, via public or private portals, as well as use it as a kind of infrastructure to do continuous mining of the genomic data that we have over time to see if we can make novel discoveries for these various phenotypes. With that, I'll take any questions.
Question. Go ahead. Hey, Ali, this is David Lim from UCLA. Thank you for your great talk. I have a practical question, actually. Are you keeping medical records and information from every individual in the cohort? So I'm not referring to molecular information, but more like medical records. And if so, how do you keep that? Is that in a repository? And the third question would be, is it retrievable for the centers?
Yeah, so we don't have their actual medical records, but we have pulled some information from their records and from surveys, really, during the enrollment process. So we do have some information. I have to look back and see what exactly it is that we have, but it's sort of mostly diagnosis status for a variety of different conditions, and then information about their family, siblings, age of death of siblings, cause of death of siblings, etc. So we do have that all available, and I believe we can make it accessible, yes.
Okay. Since you're not having what you just mentioned, having medical records from these individuals, is it still possible to go back to that, or is that, you know, like, how does that work if you want to look into more details? Because I would say that all the molecular information that you gather should, you know, should also be correlated to their, medical records, I would say, right? That's the way I look at it, but I don't know how.
Right. I'll have to find out, actually. I'm not sure if we have continuous or at least historical access to the medical records at the time of enrollment. I'll have to figure that out. I'll have to find out for you.
Yep. Okay. Thanks.
Are there any other questions for Ali?
Yeah, Ali, this is Andrew here. So I just want to follow up on the IPSC thing real quick. I mean, obviously, we've talked and really excited about the idea of using those for in vitro sort of cardiovascular assays. I guess I'm curious as to the other applications, why you were developing the IPSCs in the first place.
Okay, so yeah, so the IPSCs are being generated under this next gen grant. It came out during the ERA fund. So the goal of this project is to dissect the functional consequences of risk variance at 9p21. This is a locus that's associated with a couple of different cardiovascular conditions, but it's a gene desert, really. So there's a lot of interest to know why this region, which is a pretty high-risk region for cardiovascular disease, what is it doing that causes the risk of cardiovascular disease? So the project is generating these lines in the Wellderly individuals as well as individuals from another cohort. So there's another 75 individuals that we're generating IPSCs from that come from a what's called the gene heart cohort. These are individuals with cardiovascular disease, to look at a couple of things. So we'll be deriving these endothelial cells and smooth muscle cells and comparing, just doing RNA-seq there and comparing individuals with and without 9p21 risk status to see if we can find anything at the transcriptomic level. Of a couple of different cell-based assays will be done as well, kind of LDL uptake and similar sorts of things in the derived cells to see if there's any cellular phenotype we can see that differentiates the groups. But really, ultimately, what we're trying to do is, from a smaller number of individuals, so before that, I should say, the purpose of that grant is to generate a resource of cells to be used. So we're not necessarily using all the cells that we're generating. They'll be banked. These various lines will be banked and made available for anyone who wants to use the cells for whatever purpose. Internally, we'll be taking a smaller number of individuals with iPSC generated and then attempting to generate isogenic lines of 9p21 risk status. So this means taking the 60 kb region and swapping the risk haplotype for the non-risk haplotype, or vice versa, to generate lines that are isogenic. So the background genetics is identical, and they just differ at their risk status at this large enhancer locus, and then take those isogenic lines and subject them to these various assays that I mentioned to see if we can dissect the functional consequences of 9p21 risk variants.
Cool, makes perfect sense. Thanks. And so yeah, that, I mean, you know, the proteomics events within, you know, our center, I think tying into that makes perfect sense, as we've talked about. Perfect.
Yep.
Any other questions? Okay, thank you, Ali, for that presentation. So that concludes our presentation. Are there any questions about this conference in general? Okay, so the videos will be posted after this meeting. We'll process them, and they'll be posted online. If you have any other questions, feel free to email me or Brian. But yeah, thank you for attending this conference.
All right, we'll see you next week. Bye.
Thank you, everyone.